Hi, so now we get to talk about one of my favorite chapters because we get to talk about proteins and I get to talk about some meat and some meat proteins with you guys. So part one is going to be pages 303 to 314 all the way up until you get to functions of proteins in foods, and that we'll pick up that part in our next section. Okay, so proteins, really important because they are a huge part of what living cells are made of, especially animal living cells. So up to 60%, I believe the book might say 50%. I have it in red because I thought it was wrong. And so some cells can have even higher than what the book says. Now, let me point out, what do we mean by dry weight? So that is the weight after we've taken out all of the water. Proteins make up to 50% of what's left because water often takes up quite a bit of weight. Okay. Um, so depending on if it's an animal or a plant, it can be 70 to 95+% of the actual weight of the food product. So after we've taken out all of the water, proteins can make up to 60% of the dry weight of those cells. Now, this is a big list. I'm not asking you to memorize it, but just having a general idea, even more for your own personal knowledge than for this class, of what the protein content of different foods is. Now, I will say with this, there's multiple ways that I could introduce these percentages. I could try to do it by dry weight. Here I've made it more simple in wet basis. So the food in front of you, and I did it just as a percentage wise. So instead of taking each food and saying what a serving size is and how many grams of protein are in a serving of the food, I've said, okay, what if there's 100 grams, 30% or 30 grams would be the protein in that food. So that's a little bit misleading too, I'll admit, because you don't eat 100 grams of all of the same foods and consider that a serving. Like 100 grams of walnuts would be a ton of walnuts. Um, Also, milk, we classify that more by cups, not weight, right? Um, but just to have a general idea, so your meat, and I consider fish is technically meat. A lot of times we don't refer to it as meat, but fish, it's muscle, right? Which is the same thing as other meats are. So our meats have the most protein on a weight basis, all right, 20 to 30% protein. Um, Walnuts are substantially less. Eggs, 13%. Now, eggs are a great protein source, but there's a lot of water in them. Walnuts, the reason they're so high is that there's not a lot of water in them. Oops. Um, uh, then we have cheeses, yogurts. Then we start getting into more of the plants. Our legumes are down to like 9%. Quinoa, which is a grain or a seed, I guess, that a lot of people consider a good protein source, only has 4% protein by weight. Milk is only 3%, but think about, you're actually consuming a lot of water when you're drinking milk. And this is per 100 grams, so you're going to probably be drinking more than 100 grams of milk at a time. And then I just threw in a green leafy type of vegetable, broccoli, is also 3% protein. So the key thing here is animal sources of protein are going to be our highest amount of protein per 100 grams of weight. Okay. So now let's start getting into the chemistry of it, like we do at the beginning of each of these chapters. Talk about protein structure. So like the lipids and the carbohydrates, protein is primarily made up of carbon, hydrogen, and oxygen. But also we have lots of nitrogen in our proteins, and we do sometimes have sulfur. Um, and so we'll get to this next part in a second. Proteins can also contain iron, copper, phosphorus, and zinc, because some of these metals do bond to the amino acids that make up the protein, or bond to the proteins themselves. The basic building block, the smallest unit inside a protein, is the amino acid. And that is what this diagram represents here. Okay, so we have a side chain, which is represented by the letter R here, but it's going to contain some certain number of carbon and hydrogen atoms in some kind of configuration. This side chain may also contain that sulfur I was talking about. It can also contain more nitrogen, but it is mostly going to be carbon and hydrogen atoms. Then we have the carboxyl group. Okay, the same thing that we had in the fatty acids. Okay, the carboxyl group is what makes the amino acid an acid, just like it made the fatty acid an acid. Okay, that's the carboxyl group. And then we have an amine group, sometimes called an amino group. Okay, and this is what makes it an amino acid. Okay, an amine group is one nitrogen with two hydrogen, and these are bonded to a carbon atom. Okay, so every amino acid, every individual smallest part of a protein, the amino acids, have these three parts to it. Okay, the side chain, the carboxyl group, and the amino group, and all of these bonded to a central carbon. So I will say so the carboxyl group and the amino group is the same across all amino acids. The side chain, and why we have it just represented with an R here, is because depending on what that side chain is, determines what the amino acid is. So peptides is our next level up from amino acids. So the peptide is when two or more amino acids combine to form a bigger molecule. We're not quite at the level of a protein yet, but it's just as these amino acids are bonding. So how this works is we have two amino acids here. We have a valine amino acid, and so here's the side chain on that. Here's the amine group. Here's the carboxyl group, and phenylalanine. Okay, again, the side chain here is just represented pointing up instead of down, which is fine. The amine group on the left and the carboxyl group. And the carboxyl group bonds with the amine group. And you can see the circle here, which is where they bond. And when that happens, they release the water, okay, the H2O. So you see this double bonded oxygen is still here, right? And the hydrogen that was on the OH part of the carboxyl is now bound to the nitrogen from the amine group, okay? And the water was released. And so this bond here is a peptide bond, and we will have a peptide bond between every amino acid in our peptide and or protein. Okay, so when just two amino acids bond, we call that a dipeptide. Okay, if three or more amino acids are bonded, then we call that a polypeptide. Okay, there's no monopeptide because that would just be a single amino acid. So we have two amino acids, a dipeptide, three or more is a polypeptide. Okay. Now, so notice this peptide, it still has the amine group that was originally from the valine here, and it still has the carboxyl group originally from the phenylalanine here. So at either end, no matter how many amino acids you have as part of that peptide, at the ends, you're going to have on one end an amine group, and on the other end, a carboxyl group. Okay, so first basic building block, we have the amino acid. Next level up, we have peptides, which are multiple amino acids bonded together. And then finally, multiple peptides will bond together to form a protein. And that is how the protein is built. That's just me saying that. Okay, so how we classify amino acids, and we're going to get into this in the next few slides, just like if you're doing notes, save some space for this. There's two ways that I'm going to describe how we classify them. One is based on nutrition and nutritional needs, and the second way we classify them is based on side chains. So there are 20 amino acids that are essential to the human body, but some of these we can make ourselves by converting other amino acids within our body. And so even though our body needs them, we call them non-essential, okay? So there's these 5 here that are considered non-essential. Just like with the fatty acids though, there are some amino acids that our body cannot synthesize no matter what. And these are the essential amino acids. We must obtain these 9 amino acids through our diet. Now the book calls these, uses a different term for these. It calls these indispensable amino acids. That term is correct. If you want to use that term, that is fine. I will use the term essential because that is the term that's used more often in science and textbooks, etc., and nutrition. You don't really hear indispensable used very much to refer to the essential amino acids. Okay, now we have this other group here that is conditionally not essential, or conditionally non-essential, or conditionally dispensable, however you want to phrase it. It's conditional. And these are amino acids that our bodies can make, but in certain conditions, either our bodies stop being able to make it, or we need more of it than we can make. So a lot of times this is due to illness or stress, but it can be needed as part of normal healthy function too. So an example of when when an amino acid would be considered essential, when it's normally non-essential, would be glycine. So glycine. Glycine is conditionally essential for women during pregnancy, especially in the last part of pregnancy. Glycine is a major part of collagen, which makes up most of the connective tissue in your body. It helps hold things together. And of course, the baby's body. So there's a lot of collagen turnover in pregnancy, which means collagen being broken down and being built back up because things are growing, okay? And also fetal DNA, bones, connective tissues, their internal organs, their blood vessels, all of these require high levels of glycine. The female uterus at the end of pregnancy contains 800% more glycine than at the beginning, okay? So simply put, the demand for glycine outweighs the ability of the body to supply it. And so it must be sourced from food, especially towards the end of pregnancy, even though normally it's a non-essential amino acid. Now, luckily, glycine isn't really hard to get, but it's not a bad idea if you're pregnant to source foods that are specifically glycine rich. Okay, and then, yeah. So most of the time, these are considered non-essential, but in some periods, they are essential. Now, 3-point exam bonus question, okay? So here's a little assignment to get 3 points bonus on this next exam. If you can list the 9 essential amino acids, so these ones on this page here, okay, then you'll get 3 exam bonus points on the next exam. Spelling will count. Now, a helpful mnemonic to memorize these is PVT, which military lingo stands for private. Okay, so Private Tim Hill. This I in Hill doesn't stand for anything. That's why it's lowercase. Okay, so P is phenylalanine, V is valine. The first T is threonine or tryptophan, whichever helps you memorize it better. The other T is tryptophan, so threonine, tryptophan, isoleucine for this I, methionine for the M, histidine for the H, and then lysine and leucine. Okay, so there's a extra credit bonus opportunity if you can memorize those. Okay, so still talking about nutrition and amino acid classification. These definitions of complete and incomplete proteins. So now, as we're talking about complete and incomplete proteins, I'm not talking about the individual proteins themselves, but protein sources. We use this phrase, complete protein and incomplete protein, but really we're talking about all the proteins in a certain protein source that we are consuming, okay? So a complete protein is a food that contains all of the essential amino acids, and I should also say in a proportion that matches what we need, in a proportion that will give us enough of all the amino acids that are essential to supply our bodies with what we need. Okay, the proportion might not be exact, but eating a normal amount of this food, we would get all that we need. Okay, so pretty much any protein that is from an animal source of protein is going to be a complete protein. And it kind of makes sense because we are not plants, right? And so we need the same type of proteins to make up our muscles. Our muscles are very similar to the muscles of cattle or sheep or pigs or chickens, okay? They work similarly, right? Our organs, although they may be put together very differently, and some of our organs have very different functions, they have a lot of the same protein needs and they're made up of the same things. Versus if you think about a plant, that's nothing like what our bodies are made of, okay? And so it would make sense that animal sources of proteins are going to have the same amino acids that our bodies are made up of and need, okay? Now, there are some plant sources of protein that are complete. And so some examples are quinoa, buckwheat, amaranth, hemp, chia seeds. Notice that these are all kind of not commonly consumed proteins, so you really have to know this about them and look for them in order to have a complete protein if you're on a vegetarian diet. And I will say also, a lot of times you have to eat a lot of these things to get enough protein for your body, especially in certain states, like if you're pregnant. Now, I will say soy, if it's fully processed, it might be complete, but usually it's low in methionine, and so they often add extra methionine to things like tofu to help vegetarians get a complete source of protein while eating that vegetarian diet. Now, incomplete proteins are then therefore foods that lack one or more of the essential acids. But if they're missing one, they're not a complete protein. And whatever the amino acids there are that are lacking or missing, these are called the limiting amino acids. So whichever, and actually, so even if a protein source has all of them, if one of the amino acids is pretty low, you would call it a limiting amino acid because considering if a person ate only that source of protein, the amount of that amino acid would be kind of the limit to to what, I guess I should say, if the amino acid that they are lowest in kind of determines whether they're meeting their essential amino acid needs. It's the limiting amino acid. Um, and so if they're eating only that, you'd have to look at that amino acid and think, okay, so how much of this, I don't know, these peanuts would I have to eat to get all my protein needs? And it might be quite crazy amount because the that one particular amino acid is so low in peanuts, for example. Okay. Um, Now, that doesn't really happen in people. We typically don't consume just one protein source. But nuts and grains, these are low in lysine. Um, Beans, lentils, these are low in methionine and cysteine. So those are the limiting amino acids of those foods. Now, notice that these are just low in lysine, but these are low in methionine and cysteine. So if we eat a combination of these, then we'll be able to get all that we need, right? So grains, if we are eating, whole wheat toast and peanut butter, right? We've got the beans and lentils here that are low in methionine and cysteine, but we can get that methionine and cysteine from the whole wheat toast, and the lysine that's deficient in the grains from the wheat, we can get in the peanut butter, okay? And so the key thing there is that vegetarians especially must eat a really good combination of protein sources and be aware of the protein sources that they're eating in order to get the protein that they need. Okay, so I told you the first way that we can classify amino acids is on nutrition, and the second way is based on their side chains, okay? So you don't need to know any of these specific structures, but like I said, there are 20 amino acids in the human body, and here they are. Plus there's one extra one in this diagram, selenocysteine. So overall, amino acids by themselves are always polar. The carboxyl group here has a negative charge, and the amine group has a positive charge. But their side chains can be very different. So you see that some amino acid side chains are polar, right? We see that they have electrically charged side chains. These are polar, but they're uncharged. Okay, but some of these polar side chains, some of them have a positive charge. These ones here, some of them have a negative charge. Some are polar, but the side chains have no charge. They can lose or gain hydrogens and end up with a charge, but they can also form hydrogen bonds easily because they have structures that allow them to. So like these have the hydroxyl groups on them, and these can also, okay? And so if they can form hydrogen bonds easily, they're polar. Now, some are really important for special reasons. For example, cysteine has sulfur in it, and we'll talk about this in a little bit, but it forms bonds with other cysteine molecules, which are really strong. Glycine, it doesn't react with anything when it's part of a protein, but it has the simplest structure of all the amino acids. There's this side chain is like nothing, right? It has the carboxyl group and the amine and just that single carbon with nothing else extra on the side chain. So it's basically a side chain is just one hydrogen atom. And so because it's so small, it gives the protein a lot of flexibility for folding, which we'll talk also about in a little bit. Proline, another special one, it causes sharp bends to occur in a chain of many amino acids. And so that impacts the final structure. And then finally, some of the amino acids have hydrophobic side chains. So these are nonpolar, hydrophobic or nonpolar, and they're going to be attracted to other nonpolar compounds. So take our HDL and LDL lipoproteins. The protein part of these clusters is going to be largely composed of hydrophobic amino acids so that they do want to hold on to the lipids and cholesterol. Now, notice on the structures what everything here is made up of. So whenever we see a line, right, or a corner, right, like this, that's just carbon, right? And it's assumed that hydrogen fills in any empty bonds in these. There are no carbon-carbon double bonds unless we have the ring structures here that we see on some of them. And so whenever you see an extra line in these, hexagon or pentagon rings. So there's carbons here at all of the corners of these rings. And then the extra line means there's a carbon-carbon double bond in there. Okay. Now, if there's ever an element besides carbon and hydrogen, it is written out. So here we see the oxygen, right? We have nitrogen. We see that on all these, but there's extra nitrogen here. We have sulfur. This one that was a special one that we don't really talk about has selenium. And so we have, you can see what I talked about before. Our amino acids, which is our basic building block of our protein, is carbon, oxygen, hydrogen, and nitrogen in every single one. And some have more. And then just a couple of these also can have sulfur. Okay. So cysteine and methionine have sulfur also. Okay. So as you should have gathered from the long list of things I said the amino acids can do as far as binding, causing bends, etc., protein structure is very complex and can vary a ton from protein to protein. And there's, I wouldn't even say thousands. I would say there's probably millions of different types of proteins in nature. And the shape of this, every single protein is different, and it varies, shape and size is different, and it varies depending on what exactly the amino acid composition is. So we're switching gears a little bit, and we're going to dive into a little bit deeper into how the structure and shape of proteins is determined. So starting at the peptide level, we have the primary structure. The primary structure is kind of our protein recipe or our roadmap, depending on what analogy you want to use. It's just specifically what amino acids the protein is made of. Okay, it's just, okay, this primary structure is glycine, proline, threonine, glycine, threonine, glycine, glutamine, serine. Okay, you don't need to know all of those abbreviations, but that's all it is. Okay. It also tells us which end is the amine end and which end is the carboxyl end. We don't even see the carboxyl end in this diagram because this protein probably continues further on. These numbers here are just kind of denoting this is the first amino acid, this is the 5th one, 10th one, etc. So that's it. Primary structure, pretty simple, just the order of amino acids. Okay. The secondary structure then is what shape sections of that amino acid chain begin to take. Okay, and these shapes are going to be due to the hydrogen bonding between amino acid side chains. Okay, so the amino acids don't generally, there are exceptions, form covalent bonds with each other than the peptide bonds, but they can form other hydrogen bonds that kind of help hold their shape together. And there's 3 secondary structures that are in the book and deemed important. Um, I'm going to go in these in kind of opposite order of what the book does. So there's alpha helixes, there's beta sheets, and random coils. So the alpha helix, it is a helix. The amino acids will form hydrogen bonds with other amino acids down the chain, such that this overall amino acid chain forms a helix structure. There are certain amino acids, as you might imagine, that are more prone to this and forming those hydrogen bonds. So methionine, alanine, arginine, lysine, leucine. There are other amino acids that would never form an alpha helix because they can't form hydrogen bonds. So these would be things that are like the hydrophobic side chains, the nonpolar side chains. So examples: proline, glycine, serine, asparagine, etc. Okay. Um, yeah. So, so amino acids determine what that secondary structure is going to be, and the bonds that will be made. Okay. So the next type of secondary structure is the beta sheet. It might also be called beta pleated sheet. And again, the amino acids will determine that. So proline and glycine, both of those kind of make turns in the molecules. And you see that that is what's going on here. So you have this, and then it turns to this, and then or actually here it turned and did this, and then turned, and then this one did this and this, okay? And so then they're all like kind of lined up across from each other because of these turns. They're lined up in parallel. And then across those parallel strands, that's where the hydrogen bonds form in the beta pleated sheets. Okay, so we find the proline and glycine in those turns that are connecting the segments. Okay, and we'll sometimes see them parallel like this, sometimes anti-parallel like that. Okay. So you don't need to know this, but again, certain amino acids are conducive to this arrangement and others are kind of prevent it. Finally, we have the random coil. And the random coil, it's, hold on, I need to pause this. Okay. So the random coil, there isn't a major pattern to these amino acids, hence the word random. Unlike the alpha helices and the beta sheets. So the shape appears more random. Now it's not truly random. The hydrogen bonds and the general shape of this coil is determined by what those amino acids are, just like with the beta pleated sheets and the alpha helices. But it's not an identifiable visual pattern like the other two. So we've had primary structure, which is just the list of amino acids. The secondary structure is how different sections of the amino acids form a certain shape. Then we have tertiary structure, which is the overall three-dimensional structure of an entire amino acid side chain. Okay, so we have within like a huge side chain, or sorry, not side chain, amino acid chain. So big polypeptide. Um, so within this big polypeptide, we have beta sheets, we have alpha helices, we have some random coils, and then overall, what is that three-dimensional structure? And there's two main three-dimensional structures that we see. One is globular, so it's just more globe shaped, circular shaped. It may not have a identifiable form shape to it, okay, versus fibrous. So fibrous is going to be things that they look linear. Now, the amino acid chain is not linear, but it's going to be situated in such a way that overall, that when you look at it from, you know, a far back view, it um, the structure of that polypeptide looks fibrous. It looks linear. Okay. So let's go into each one. So globular proteins are often, though not always, proteins that will be transported around the cell or body. Um, if they are stationary, they're generally going to stay in that one spot to form a specific reactionary function. Their purpose is not to form networks with other proteins and make structure. Their purpose is some kind of reaction that they are helping along. Okay. So the first example in the book for globular proteins is hemoglobin. Now, hemoglobin is actually composed of 4 amino acid chains. And so you can see them here, 1, 2, 3, 4. So the tertiary structure is not referring to all of this. It's just referring to a single chain, okay? And I'll get to this in a couple slides, okay? Um, but each amino acid chain of hemoglobin is globular. So this is not, when we're talking about tertiary structure, just keep in mind, it's not necessarily the whole protein yet, which the book doesn't explain well. Now, hemoglobin is the protein in our blood that is used to transport oxygen. So that's what this little disc kind of shaped representation is here, is this is the heme group, and that's the center of the domain, of each domain of the protein, okay? And that holds the oxygen and releases it when it gets to parts of the body where it needs to be released. Now, the book also gives the example lipoprotein. I'll say lipoprotein is not a good example at all, because lipoproteins actually have more than one protein on the exterior of the whole big cluster of lipids and proteins. So X that from your memory as you read that, because that is not lipoprotein. Each individual protein in the lipoprotein is an example of a globular protein, but lipoprotein itself is not a good example of a globular protein. Now, the examples of casein in milk and the albumin in egg white are actually good. Casein, for example, is found in milk. It provides a source of essential amino acids for the newborn, and it's also a carrier of calcium and phosphorus. And it's also an emulsifier, helping to mix the oils, fats, and water in milk so that they're not all separated when it's being provided to the infant. Okay. And we'll talk more about casein in some future slides, because casein is some good examples for some other things. Okay, so then that was globular. Now fibrous. So fibrous proteins are often very heavy in helix-shaped strands. Because you think about helix, that's got that kind of overall linear-looking structure if you're looking at it from, you know, a bird's eye view. Fibrous proteins are often structural. So collagen, which in this diagram is the black lines, and elastin, which is the kind of squiggly off-white lines here. These are both connective tissue proteins, and they form the structure of muscles, our organs, our skin. And so they're fibrous. They form networks, okay? Keratin is the protein that forms the structure of hair, skin, nails. And so structure. Now, myosin isn't really a structural protein per se. Myosin is part of muscle, but it's also not fully fibrous either. So you see myosin, okay? It has the tail here that looks fibrous, and then a globular head. Okay, the fibrous part of the myosin and muscle provides strength. And fibrous proteins tend to do that, provide strength. And then a globular protein part of the protein. So notice this is actually all one protein, but this is parts of the protein. Okay, so the amino acid chain that is at the end here is globular, and the globular part provides the binding and the action. So the way muscle works is myosin will, and this is very simplified, but myosin will bind to actin, and then once it's bound, it moves it. So it binds it, and then kind of like a lever, it moves. Here's the actin, here's the myosin, and it moves it. And that is how a muscle contraction occurs. So the globular protein is the action, the binding part, and the fibrous part is the strength behind it. Now, in general, fibrous proteins link together, form strong networks, and multiply their strength further. Okay, so you can see like there's networks of the collagen and elastin, and so that's stronger than just one collagen and elastin, or even if they were all just lined up like this and not crisscrossed and stuff. Okay. Now I will note here, actin here, it looks like a fibrous protein, but it's actually a strand of many globular proteins. Each one of these right here is it's its own little globular protein. Okay. So the book does not go farther than that. It stops at tertiary structure. I'm going to go one step further and talk about quaternary structure, because like I said, the book treats tertiary structure like the protein structure, and that is not the case. The actual overall protein structure is the quaternary structure, and so that is the structure of the entire protein. I say more than one amino acid chain. It is the entire protein is what quaternary structure is. Okay, so please add this to your notes and understand this. So like I was talking about hemoglobin, okay? There are 4 globular domains of hemoglobin, and they combine to form a quaternary structure that is also globular, okay? But the tertiary structure is just each single one of these parts of the hemoglobin, and the quaternary structure is the entire protein, okay? So the hemoglobin then we know has 4 heme groups. Okay. So now let's get into the molecular interaction of proteins. We've already talked quite a bit about hydrogen bonds, okay? They're essential to forming the secondary protein structure. We've talked about alpha helixes and beta sheets, right? But they're also essential to forming the tertiary structure. So go back a little bit. So like this, like how is this gonna all be kind of connected and form that globular protein, okay, and the shape of that overall globular protein? They determine how the protein molecule folds and how it maintains a specific shape. Also, amino acids that have not formed hydrogen bonds within the protein structure, but do have the ability to, they have a hydroxyl group. These amino acids can then form hydrogen bonds with other substances, such as water. So like these are the 3 amino acids that have hydroxyl groups as part of their side chain. They have the ability to form hydrogen bonds with water. And so proteins that have high enough levels of these 3 amino acids are much more soluble in water. So this is why casein is suspended in milk and doesn't just float at the top or sink to the bottom, for example. This is why whey can be mixed into drinks. Okay. Another molecular interaction of protein is disulfide cross-links. Now, what this means in real language, not less sciencey language, disulfide to sulfur. Okay, cross-link, another word for bonds. Okay, so two sulfur bonds, and so disulfide bonds are covalent. They are very strong. They're not easily broken. And this provides some major stability to a protein that has disulfide bonds as a part of it. So like I said before, there are two amino acids, cysteine and methionine, that both have sulfur in them and have the ability to form these disulfide bonds. Now, when these disulfide bonds are broken, we get like some free sulfur action going on, and so that's when we get rotten egg smell. And that's exactly what rotten egg smell is. There's lots of cysteine and methionine in eggs, and if those bonds are broken, because these cysteine and methionine are going to be making disulfide bonds within the proteins that they're part of, and those bonds are broken because of protein degradation, because of bacteria, eating away at them, then you get the rotten egg smell because you have that sulfur that is no longer part of the bond. Okay, so other molecular interactions include hydrophobic interaction. So hydrophobic means fear of water, water fearing. And we've talked about hydrophobic before, but maybe we've talked about it more referring it to nonpolar, right? So nonpolar molecules are hydrophobic, they're water repelling, and they're lipid loving. So the side chains that are nonpolar on an amino acid are hydrophobic, same as oils. These side chains hate water, but they love lipids. So an example here, casein, again. So casein actually has several forms. Kappa casein is hydrophilic, but alpha casein is hydrophobic. And so what will happen is kappa casein kind of sits on the exterior. All the caseins kind of merge together, and kappa casein sits on the exterior of this little globule of several casein proteins that are formed together in what's called a micelle. It's just a large globular structure, and all the hydrophobic proteins are on the interior, and the hydrophilic parts are on the exterior. But if we do something to remove the kappa casein, then now we have hydrophobic parts exposed. And we have casein that does not want to be mixed with the water. So then it will more readily combine with the fat in the milk. It will also combine with even more casein molecules and coagulate to form cheese. Okay, I think this might be the last molecular interaction. If not, I'm sorry. Okay, color changes of protein pigments is another one. So now we get to use a meat example, which I'm excited about. Okay, so let's talk about myoglobin, which is what this whole example is about. So myoglobin, the name sounds an awful lot like hemoglobin, right? And the structure is quite similar, but myoglobin actually just has one domain. All right, the hemoglobin is composed of 4. The myoglobin only has 1, and so it just has the one heme group. So the function of hemoglobin is to take oxygenated blood to the tissues, drop it off at the tissues, and then go get more oxygen. Myoglobin kind of has the reverse function, right? It its function is to grab the oxygen that hemoglobin has gotten or has given, and then give the oxygen to whatever cell processes need oxygen. Okay. And so myoglobin, because it's very similar to hemoglobin, it looks a little bit like blood. Okay. And it is responsible for the red color of meats. The more red a meat is, the more myoglobin it contains. Right. So like a chicken breast contains a lot less myoglobin than a beef steak. Okay. So this heme group is affected by what is attached to it, and also what its oxidation state is. So I'm sure you've probably heard that blood with oxygen is red and blood without oxygen is blue, if you've seen some bad diagrams and just made the assumption from anatomy books. But really it's like a darker red, maybe somewhat purplish. Okay. Same kind of thing happens with myoglobin. When the myoglobin has oxygen attached to it, it and the iron is in a reduced state, then it is oxymyoglobin, and it is a nice bright red color. If it loses that oxygen, but the iron is still in a reduced state, then it turns a little bit more purple. All right. Purple isn't a great descriptor, but you can see it's a darker red, a little bit purpley colored. Okay. It's what you see when you have like a vacuum packaged big piece of meat. Okay. Now, if that meat has been out for a long time, it's starting to get old. Think about what I showed you the other day with the meat that had been in my freezer for over a year, and it had brown parts to it. That is met myoglobin. Number one, it may or may not have oxygen on it, but the iron has been oxidized. Okay. So there are some corrections here from as far as definitions that are from the book. Okay, the book is confusing the terms oxidation and oxygenation. They are quite different. Okay, so the book says oxidation is adding oxygen. That is absolutely positively wrong. Okay, oxygenation is adding oxygen. Okay, deoxygenation is removing oxygen. Okay. Oxidation is removing, let's just say an, not and, removing an electron. Okay. We remove an electron from this iron, making the charge on the iron more positive. Okay. That is oxidation. Okay. And reduction would be the reverse of that, adding an electron. Okay. Now I'll also kind of point out here, there's a fourth one that I kind of skipped over. This is carboxymyoglobin. If we add carbon monoxide to the myoglobin, we also get a bright cherry red color, and this pigment is called carboxymyoglobin. And it's actually more stable than the oxymyoglobin, but it can oxidize also. And we do see this in the food that you buy today. A lot of times the meat you get from Walmart, that it has the wrapping isn't like touching the meat. This is a modified atmosphere package. It was not cut and packaged at Walmart. It was packaged at some distribution center. And they actually insert a very small amount of carbon monoxide gas into the package. And you can see like there's what we call a headspace in the industry, where there's air between the meat and the plastic. If anytime you see that, it's a modified atmosphere package, and it may or may not have carbon monoxide. Sometimes they use some other things to keep the meat. Sometimes they add like extra oxygen to do it, but anyways, a small amount, very small amount of carbon monoxide. It's not dangerous for you to open the package or anything like that. Will also turn meat bright cherry red. Okay, now continuing on with more meat, because I know the only reason I do this is because the book does talk about it. So sorry, here was just another visualization of the exact same thing that was on the previous slide. But why is meat pink when it's cooked, right? It's the same idea, it's just different compounds. So with our cured meats, like our ham, our hot dogs, etc., nitric oxide has been added. We usually add that as far as ingredients in the forms of nitrites, and then the nitrites is converted in a chemical reaction. It's kind of on its own to nitric oxide, and the nitric oxide binds to the iron. So maybe I didn't say this before, but the oxygen or carbon dioxide is actually binding to the iron in the heme group. I think, I don't know, it's binding to the heme group. Actually, I think it's not bound to the iron, but it's bound to the heme group. Anyways, so if you have water bound to it and there's no oxygen, this is the deoxymyoglobin. This is the purple one. We add oxygen, we get the red. We take out the oxygen and we have water bound to it again because oxygen can't bind to it anymore because we've oxidized it. It's the metmyoglobin. If we are starting out with the deoxy, we add nitric oxide. We'll have nitrosomyoglobin, which is not very stable and it's easily oxidized to this metmyoglobin. But when we cook it, then we get what's called nitrosohemoglobin, which is a little bit lighter in pink color than when it was raw. But that's our cured meats. Now this can still oxidize. It's not as easily oxidized, and you get more of a gray color with this metmyoglobin because it's cooked versus the raw. So lots of neat stuff. Okay, so denaturation of proteins. So switching gears now. Denaturation is any change in the shape of the protein without breaking the peptide bonds. We can break the hydrogen bonds all we want because that's the only way we're going to change the shape, but without breaking the peptide bonds. So we still have the exact same amino acids in the right order configuration. Coagulation is a permanent denaturation. No, not all denaturation is permanent. If it's just denatured a little bit, it might be able to reform hydrogen bonds and bend back to its original configuration. But some forms are permanent. So coagulation is when you have a liquid or semi-liquid protein and it has formed clots, basically. So great example, easy example is egg whites being cooked, okay? What actually happens in this irreversible denaturation is, so egg whites are made of albumin, okay? So they denature, they unfold. They were this pretty globular shape. Now they unfold into something a lot more random. And then with heat being added, you form new cross-links between the proteins. So there weren't cross-links before, but now we get cross-links between the proteins. And so that's why they are now one solid substance and not liquid anymore. Then this example is cheese making and coagulation that occurs in that. Okay, so with denaturation of proteins, we will typically get loss of solubility. So any side chains that are hydrophobic in a soluble protein, right? Well, you bend this or unfold this protein now, and now those hydrophobic side chains, which may have been bound with something else or were kind of enveloped inside the protein and kind of on the interior are now exposed. And so it makes the protein itself more hydrophobic and less soluble in water. And so what will happen is you get precipitation of the proteins comes out of solution, and sometimes water loss. So this example here is like ground beef, right? If you cook ground beef, you'll notice that you get a lot of liquid coming out of the ground beef. Some of that is fat, but a lot of it is going to be water. And it's not necessarily, nobody added water to your ground beef or anything. It's that those proteins, as they're denaturing, are less able to hold water than they were previously. So how are proteins denatured? I mean, cooking is a way that we very purposely denature proteins. Um, and something to understand about that is um, like the rate of chemical reactions increases for every amount of temperature that you increase where that reaction is taking place. Well, with protein denaturation, that rate is increases by 600 times for every 10 degrees temperature increase. That's way more than other reactions. And that's because there's very little energy that is required to break hydrogen bonds. Cold temperatures can also denature protein, not as severely and not as often, but it does happen. So here's an example. So this is raw milk, and you freeze it, and then when you thaw it, it looks like this. That's because the proteins have been somewhat denatured. You freeze egg yolks. So this is kind of a diagram of fresh egg yolk and the protein bonds that are forming. Frozen, you can see that there's some more coagulation, um, and the lipids are more, I guess, the proteins have kind of coagulated and the lipids are just more dispersed. And then uh, after you freeze it a long time, so this is a short time of freezing, long time, you really get those lipoproteins really bonded together, more coagulated, and they've actually formed a bit of a gel. Okay. We can also denature proteins through mechanical action, so beating, rolling, kneading. So what kneading actually does, so we have gluten proteins in flour, we add water, and those gluten proteins form long squiggly strands. Well, then when we knead the flour, knead the dough, those gluten strands are stretched out and they form this strong network. All right. Now, too much mechanical action leads to a network that is too tight and too tough, and you get bread or whatever it is that you are trying to achieve that is too tight or too tough to eat, okay, or hard, right? And so there's a certain level of denaturation and mechanical action that you want to achieve with that, and sometimes want to avoid. Um, Other physical methods, the book mentions sound. This is very uncommon, so I'm not going to even really go into it. But irradiation. Um, Irradiation is not really used in meat commercially, um, and it's used a lot in spices, but there is not a lot of protein to get denatured in the spices. But in meat, there is. And so if you irradiate meat, you get color changes, you reduce the meat tenderness, and you reduce the ability of the meat to hold water because it is denaturing the protein. And so this is a picture from a scientific study of irradiated and non-irradiated meat. And so this is one of several reasons that this has not taken off in the meat industry, is because you get this happening. And that's not appetizing, and it's affecting the quality of the meat, not just how it looks. Also, there are chemical methods of protein denaturation, so pH, adding acids or alkalis. So examples here are denatured soy protein. So the soy is put into an alkaline solution, and then put in an acid bath, and the end result is this protein that kind of looks like meat. And so you'll actually see it on an ingredient label often as textured soy protein. Um, y'all aren't as familiar with it because we're homeschoolers, but school cafeterias use this a lot. They'll often have hamburger patties that are not just hamburger patties. There's also some soy mixed in with them to, I guess, decrease the cost. And so I lovingly refer to this as kind of like school cafeteria hamburger mix. Um, so also can add acid to milk to get sour cream, yogurt, buttermilk, and then poached eggs. If you add a little bit of vinegar, it makes a better poached egg because in theory it prevents the I guess it causes denaturation to happen faster, so that egg kind of stays more in one place and doesn't spread out throughout the hot water while it's denaturing. We can also use salt as a chemical method of denaturation. Now, the book talks about how salt can toughen legume proteins, and there's some conflicting things out there, so I'm not going to really follow that. The salt actually softens the skin of beans, and it may allow less water penetration, but it also leads to less split skins because of it softening the skin. So this was an example. It might be hard to see it, but if you look at this one that is unsalted, all of these beans are split. And so yes, this might be a little bit firmer, but the person who did this said the texture was still great. Um, it it didn't get soggy because the skins had split. Okay, so take what the book says about the legumes with a grain of salt. Aha. Meat binding. This is actually a better salt denaturation example. So in sausage production, salt is used to extract and cause binding of meat proteins. So this is how a hot dog stays together as one solid piece. Okay, the salt plus agitation plus then eventually heat when you cook it equals coagulation and binding of the meat proteins. And so, I mean, you look at this picture, this looks sticky, doesn't it? And that is because of the extraction of the proteins with salt. Now, too much, if you're talking about meat, too much of any of these, of the salt, of the agitation, or of the heat, would lead to a tough, rubbery product. So you have to control how much denaturation you are having occur to get the product that you want. So we went a little bit longer with this. Proteins have a lot of information. I hope everyone has had or will have a wonderful Christmas break, and we'll see y'all soon.